Session on human biology. I'm very pleased to introduce Paul Newcombe from GSK, who's going to tell us about causal machine learning for biomarker subgroup discovery in randomized trials. So let's welcome Paul.
Thank you. So just a quick disclosure there. Probably obvious. When I moved from academia to industry about 6 years ago, they were starting to more frequently measure high-dimensional multi-omics in clinical trials. At that time, really just phase 2s. And over the last 6 years, I've seen that now increased to the point where I think there's 5 or 6 phase 3s at GSK this year where multi-omics will be measured. So it's becoming far more routine. So we're talking here about things like proteomics, transcriptomics. This, of course, is in one part driven by falling costs of these assays, but also new technologies like Olink, for example, allows the quote-unquote affordable measurement of proteomics at large scale, really for the first time. And there's a whole bunch of reasons for doing this. The common aim of these efforts is nearly invariably to try to identify baseline predictors of efficacy, so of treatment versus placebo benefit, and that might be to understand why a trial has failed, influence designs of future trials, influence what new targets we might try to go after. No treatment will work for every patient, so if we can understand which patients an existing treatment doesn't work for, it might determine where we focus next. So statistically speaking, to identify a biomarker that is predictive of treatment effect, we're talking about a interaction test, a statistical test of the biomarker treatment allocation interaction. But it's increasingly well recognized that to do that exhaustively across hundreds or potentially thousands of biomarkers is highly suboptimal. Two main reasons. One is many univariate one-at-a-time tests followed by multiplicity control is very conservative, and these sorts of analyses are very underpowered to begin with. And another reason is it's surprisingly easy, and there's a really nice reference that will explain this far better than me at the bottom. That's the Lipkovich review from 2017. That is still very, I would say, very surprisingly current in the methodology and questions it discusses. But it's surprisingly easy to construct simple examples involving just 2 or 3 features of a truly nonlinear or truly combinatorial effect on efficacy that would be missed by one-at-a-time testing. And it's not to naively say that the component features would look completely inert in terms of predicting efficacy, but they might rank poorly in the context of thousands of tests. The good news is, over the last 5-10 years or so, quite a lot of new methods have appeared from really the intersection of statistical causal inference and machine learning, which aim to repurpose some of the high-dimensional ML techniques to this task of predicting treatment effect. But I think it's fair to say, if anybody on the room knows differently, I would happily have I would happily be put right on this, but I think it's fair to say these methods are largely untested for the problem of characterizing predictors of efficacy from high-dimensional omic panels in trials. And that's largely because a lot of this sort of data is still new in industry, and it takes time for these sorts of data sets to slowly permeate out into the wider world, and therefore inform sort of methodological assessments and so on. So the initial aim of this work, and I'm going to say up front, some of this is already out of date, so apologies. And again, I wouldn't paint myself as an expert in any of these approaches, just an applied practitioner, so please don't hold back on corrections or anything we might have missed. But the initial aim of this effort was to try to evaluate some of these newer approaches for detecting biomarker signatures of efficacy. So the first task was to decide what we were going to evaluate, and just want to give credit to Ashwini here in the team. She joined us from the Turing Institute a few years ago and had some experience applying these so-called causal machine learning techniques. This is out of date now, and apologies, I just realised that yesterday. I do need to update this slide, so there's several methods that should be on here and aren't. But at the time we did this, we decided to bucket approaches into these three groups. So each of these three methods is selected to represent a broader class of type of approach, and that would be sort of sparse regression type approaches, the modified covariate method. X-learner is representing here the wider class of meta-learners. It's not what I would have chosen today, to be honest. I would have gone with the DR learner, but we'll come back to that. And causal forest is an adaptation of random forests. So I'll now just do one slide on each of these methods, just to give you a sense of how they work if you haven't come across them before. So starting with the modified covariate lasso approach, the traditional approach to trying to identify a feature that interacts with a treatment effect. So just to reiterate, we're considering the problem here. We've measured a bunch of stuff at baseline in a randomized trial, and we're trying to understand what at baseline is predictive of future efficacy. And efficacy here I'm defining as the treatment versus randomized comparator benefit. So that would be a predictive biomarker as defined by the FDA. So just want to clear, because the word prediction obviously can mean lots of different things in the field of statistics, but in this case we're talking about predictors of efficacy. So as I said, traditionally, statistically, we test that with an interaction test. That's the model at the top. It's surprisingly complicated to extend that testing framework to high dimension. There's a nice reference towards the bottom there that goes into detail about why that is. It's not really a conceptual problem. We know conceptually how to extend that model to have many, many other features involved. It's a computational problem, and the problem of trying to search over sort of hierarchically linked groups of covariates, so the main effects and the interactions between them. So this approach, which is actually quite old now, employs a simple trick where you basically take your covariate matrix and multiply it by essentially the binary treatment allocation, and that gives you this Z matrix. I have to ask for a leap of faith here. I don't find this particularly intuitive, but it is quite back of the envelope mathematics stuff. If you replace your covariate matrix in your regression model with that Z, the associations you identify correspond to the interaction terms in the expanded model at the top of the slide. So the really sort of compelling thing here is that we've turned a interaction testing problem into a far simpler prediction modeling problem, which means that we can apply any off-the-shelf ML or statistical learning procedure which has been developed for identifying predictive main effects as opposed to interaction terms. There's no such thing as a free lunch, as they say, so you do lose efficiency compared to the formal model at the top. The authors propose a mitigating approach to that, which we've explored using, where you basically, in a first stage, fit a prognostic model and incorporate that into the second stage when you actually do this. What else? There's another problem with this that escapes me now. But just to also note that we have efficiency augmentation refers to this two-stage approach I just described, where you bring in the prognostic information. For error control and feature selection, just to note, we've combined this with complementary pairs stability selection, which was developed by Rajan Shah, who's based here, I believe, and Richard Samworth, which is a way of allowing a sort of quasi-FDR control for really any variable selection algorithm that gives you in or out decisions on each variable. You basically bootstrap many times, reapply the variable selection procedure, and look at the selection probabilities across the bootstraps. But this gives you some theoretical results for the error control in the false positives if you were to threshold at different levels on those probabilities, selection probabilities. So the next approach we'll talk about is causal forest. So I imagine many of you in the room are familiar with random forest, but just a quick recap in case anybody isn't. So on the far left of the slide is a cartoon regression tree where you have a series, a set of variables ordered with thresholds that partition your data set, and predictions from this regression tree will be based just on the average outcome values in each partition this creates. To fit that tree, you would optimize for a classical model fit criteria like minimizing residual variance, for example, to decide which features and in which order and which thresholds. So fitting a single regression tree has generally pretty terrible out-of-sample predictive performance. But if it turns out that if you do this many times, so you bootstrap your training data many times, fit many regression trees, an average prediction over those trees, and this is the so-called random forest approach, that can offer very good predictive performance indeed, and in fact, many of the earlier sort of open machine learning challenges were won by variants of the random forest. But out of the box, this is a tool for predicting an outcome from a set of features, not a tool for predicting a treatment effect, a parameter. But it would be great if we could use this sort of framework for efficacy prediction, and that's what the causal forest method offers. One of the key contributions is a different type of model fit criterion you're optimizing for, and it's basically you're trying to optimize within each of the branches, within each of the partitions. You would calculate a treatment effect, and you were trying to optimize for maximum variability in that treatment effect across the partitions. Hopefully that's somewhat intuitive then why this works, because it means you're explaining variability in the treatment effect. So the third method, which as I say you could think of as just a generic representative of meta-learners. So these techniques combine the notion of potential outcomes from causal inference with machine learning methods for estimation of treatment effects. What do I mean by that? So the yellow box tries to give us sort of a simple recipe for what is done. you would train a predictive model in each treatment arm of your trial for the outcome. So at this point, you've got a predictive model for the endpoint or clinical outcome under treatment, and another one that predicts the outcome under control or placebo or standard of care. For that step one, it's important to note that's a standard predictive modeling task in each treatment arm, so you can use any off-the-shelf machine learning or statistical learning or AI algorithm. You then, in the second step, use those models for each patient to impute their potential outcome, which is the outcome we would have observed under the treatment they weren't given, the alternative treatment. And then at this point, for each patient, we've got a pair of outcomes, one under the treatment, one under the comparator. Obviously, one of those is imputed, and we can, but it means we can impute their personalized treatment effect. So now for each patient, we've got their individual treatment effect imputed, which is a number. So then in the third step, we do another run of predictive modeling where we regress those imputed individualized treatment effects, again on the baseline biomarkers. And again, at this point, we can use any standard off-the-shelf machine learning software because it's a classical predictive problem. And in the end, we build a predictor of individualized treatment effects. It will be overfitted because we've used the data twice to build these imputation models, and then again to impute to model the imputed treatment effects. And the authors in this space are very knowing of that data reuse, but as I'll come to in a bit, despite the overfitting, these things can still perform very well. And the way you assemble these pieces for the final predictive model in step 4, there's various ways of doing that, and the different papers in this space offer different, or argue for maybe slightly different ways of going about that. And just a caveat that at the time we did this work, we weren't sure how to combine this with variable selection. That is a fast-moving space in this field, so we haven't used this for feature selection in this work, but that would be possible these days, and it's something we need to look at. So this is just a quick recap. I think the main thing I would point out here is that the modified covariate lasso compared to causal forest and X-learner. So we've looked here at X-learner with random forest. The authors look at combining it with random forest and BART for the predictive steps. So fundamental difference between the X-learner, causal forest, and modified covariate lasso is whether they are designed for capturing non-linearities in the feature space. The modified covariate lasso is basically a high-dimensional, simple regression model. There's just main effects for each feature. Just one slide on a really simple simulation study we did. So we took a sort of an architecture from the modified covariate lasso paper, which was the only one of these papers that actually explored a high-dimensional setting in the paper. So that was 1,000 features. So at the time we were thinking about O-link, proteomics and panels which might have 1,500 proteins on, and we might expect to be analysing about 1,000 RTQC. That was the thinking here. There's 4 truly predictive simulated features. Some of them have prognostic effects as well. 2 do, 2 don't. We simulated 2 scenarios, phase 2 and phase 3, basically just by altering the N for the trials in the disease area we were thinking about at the time. Those N's correspond to the sizes of those phase 2 and phase 3 trials. And the level of variation explained by the model, and this is hugely arbitrary, but there are variants on some genes that might explain up to 15% of variability in the protein product of those genes. So we were going for 10-15% variation in the overall outcome, but it's feel free to dispute that, completely arbitrary. So just going through the results quickly. On the left-hand side, we're evaluating feature selection, and on the right-hand side, we're evaluating predictive performance. So basically, the modified covariate lasso seem to do quite well for feature selection, which is the green box. It seemed to identify most of the true predictive features while controlling error. I think that indicates that I've probably made this problem too easy, because I don't think I would expect this procedure to find all true predictive features in reality in a phase 3, let alone a phase 2 trial. But moving to the right, so here in the simulations, we use one simulated data set to train a model, and then we apply the implied stratification to a second simulated data set and simply test for an interaction between the predicted stratification and treatment in the new data set, and the y-axis is the log 10p, minus log 10p value of that interaction test. And what's interesting here is that I think what's interesting is the meta-learner, the X-learner, does really well, particularly at phase 3, as in very consistently is leading to a significant predictive stratifier. And just again to reiterate, that's despite the fact it is overfitted in the training data. It still beats, in my view, the procedures that aren't overfitted technically. So I'll stop there on the simulations. And just give you some results from an application. So this is in lupus. So lupus is a horrible disease. It's an autoimmune disease. It can affect pretty much all parts of the body. Belimumab is a GSK treatment that was licensed in 2011. It targets BLyS or BAF, which is a protein involved in the life cycle of B cells, which is a white blood cell, and there's a theory that this is overexpressed in lupus patients, and there's a theory that these B cells are naturally dying as they should, and so end up attacking the body. So this treatment targets those. 52 and 76 is a pooling of the original pivotal trials that were used for filing, and baseline transcriptomics were measured in a subset. Need to be clear, only a small portion of the overall trial patients have been assayed here, so this is very much, in our view, a proof of concept. I wouldn't take anything I'm about to show you requires validation, but there is interest in the company for exploring what is characterizing heterogeneity in response to potentially influence new treatments in this area. What aren't we capturing with belimumab? So the computational biology team at the company had done one at a time interaction tests across, I think it's 1,500 transcriptomic gene expression probes after QC, and none of them were significant, even with a very liberal FDR control of, I think we even explored up to 50%. So we were asked to try applying some of the causal ML approaches. We focused here on modified covariate lasso because the goal here, I think, was to understand as opposed to predict. So we're much more interested in feature selection, what genes may have been driving the differences in efficacy. And interestingly, 5 genes came up, significant, with a reasonably liberal FDR control of 10%. So this I've been asked to redact, so you'll have to take my word for it, but of these 5 genes, 4 of them are plausibly linked to the mechanism of action of the treatment. We don't have transcriptomics in another randomized trial at the moment of this drug. We do have it from this Bliss-Believe study, which was a superiority study against a combination with a competitive treatment. So we attempted a partial validation. If these genes are predictive of efficacy, you might expect them to be predictive of response under treatment. And of the 4 candidate genes, 2 of them were significantly predictive of response in this other trial. So I'm going to whiz through a bit to allow maybe a little bit of time for questions. This was a basic exploration of whether this is clinically useful. So we take the 2 genes that were significantly predictive of efficacy and response in this second trial. And in honesty, I don't think this is going to be clinically useful. And I'm quite interested in better ways of doing this sort of evaluation. But it looks like to double response rate, we will be looking at removing about half of the target population, which is not what commercial wants to hear, but also it's denying patients a treatment that might work for them. There's not just a commercial perspective here. So this is a case where there might be something significant going on, but the predictive accuracy, I think, is just too low to be of any utility. So just to wrap up, on the left, I think from our simulation study, there's more potential here in these approaches applied in high dimension than, to be honest, I was expecting. And we've seen that borne out now in a range of programs. So we've now applied these techniques to 4 other programs at the company, and we're increasingly applying it to multiomics in those programs as it's being generated. I'll stop there. Thank you very much.
Can you hear me? Yes. Thank you very much, Paul. Does anyone have any questions? Yeah.
Are biomarkers like let's say that hence you have a bit more confidence with the causal relationship, or I'm struggling to see how is this prediction has been
So can you repeat the question? People online.
I think yes. So the question is, how is this causal inference? I meant to say that at the beginning. I think I'm a bit of a charlatan today. This isn't really statistical causal inference. So in a randomized trial, of course, the effect of the treatment we can interpret as causal. Most of these methods are designed for application to non-randomized data. Here we are applying them to randomized data. So all of the clever machinery for learning propensity scores and so on, we're not actually employing.
Yeah.
So I guess you're getting at the fact that lasso can give very spurious results when applied to highly correlated features. So we will prune heavily. So we want to prune down to maybe maximum correlation 80% or something. That, in my view, is unlikely then to lead to numerical issues during the search, but we will keep a careful log of what we've pruned out. So maybe we choose one representative for 20 features. If that representative comes up, then we go back to the other 19 and try to figure out which might be the most likely explaining gene.
Yeah, Ching, your last question.
If one the other one to find biomarkers, and these are not quite the same, right? So I'm interested in your take on that and designing
I mean, thank you, because that is a very important aspect of this sort of work, and it's something we work hard on communicating in the company. There are sometimes very practical reasons.
Repeat the question, please.
Oh, apologies. Yeah. So it was, I think, a general prompt on the sort of dichotomy of aim here of wanting to predict efficacy versus identifying the features that are causing differences in efficacy. So you might want to predict efficacy if you're designing, for example, trying to make the next trial more efficient, and you want to recruit patients into that trial who are more likely to show a response. That could be, yeah, there's many reasons I'm sure you can imagine you might want to do that. But if you want to understand which genes, pathways, proteins, and so on are involved in the efficacy, the predictive performance may be irrelevant, because you're not actually trying to use those to predict who will benefit. You're trying to learn. I mean, something that surprised me coming into big pharma was often how little is understood about the mechanism of action of treatments. So sometimes it's just expanding basic understanding of why does the drug work and in what patients does it work in. But it could be to inspire or to inform the development of new treatments. You're quite often in a situation in big pharma where you've got something being trialled at phase 3, has gone to market, and say 10 other assets coming up very early in the pipeline, just going into human trials. So you want to understand how they would complement your sort of blockbuster. So again, understanding what patients respond to what can inform how you might prioritize what's starting to come up.
Okay, thank you very much.